I want to say, so I am heavily involved in single-cell genomics. I have been, not that I am a scientist who's running the lab, who's doing it, but I have a lot of collaboration trying to apply those new techniques into the clinic. I remember a few years back when we were presenting our data on the single cells, everybody was like, oh, there's only a few labs who can do it. If you look at it now, we're at the point that we can actually utilize the single-cell genomics on the more wider network of trials and specimen analysis, meaning that these high technology assays can be actually generalized, and which is even more important than bringing a new assays. For example, even though using the single-cell genomics, now we're at the point to the next level of single-cell transcriptomics and spatial transcriptomics. So in addition to know what genes are overexpressed, what proteins prototype is overexpressed, we can actually know which cell and where in the geographical area. So like, for example, Dr. Blanchini, presented the data on the NeoTrip where he actually using the imaging mass spectrometry, where it's not exactly a spatial transcriptomics, but you can actually spot where and how much expressions then each cell actually has. And so I think those type of technique will be more and more utilized. At Baylor College of Medicine, we also study something we call proteogenomics. So in addition to DNA, RNA, we also look into the deep dive of the phosphoproteins and how this effector of the proteins can actually make a difference in their behavior of cancer, how we can actually target them, segregate them into the different, behaviors of cancer. So more to come.